And Edith now will share with us what she's doing, which I think relates in turn to predictors of prognosis and outcome, right?
Yes, a true pleasure to be here today, to see many friendly faces, and to have met some very nice new people who I hope may become friends in the future. Certainly also an honor for me to be part of this esteemed group today, and to talk to you about some of the work that we are doing, which I'm going to divide it briefly into three different areas. The work that we're doing at Mayo Clinic per se, the work that I'm doing at the national level, and then take this to the work we're doing at the international level, all of which is partially funded by the BCRF. So I must thank you for your contributions to this date and for further contributions. So at Mayo, what we are concentrating on is the understanding on the unraveling of the genetic or genomic makeups of breast cancer, because we understand that the state-of-the-art treatments that we have today are possible because of the work that was done in the past several years by scientists as well as patients who participated in that research. So we are right now taking specimens from patients, as Dan alluded to, and we are performing state-of-the-art genomic analysis, which is called next-generation gene sequencing. So just by that term, you realize that this is really the latest thing. And that technology allows us, in just a few days actually, to get a printout by a machine of the genes in tumor cells, and maybe 24,000 genes. In addition to that, we can get a printout of how the genes might have been altered, because alteration of genes may have an impact on the product of the genes, which are the proteins. So we started doing the work in cell lines, then we started doing work in human specimens, because patients allowed us to have the extra tumor specimens that were not needed for diagnosis to be able to do this innovative work. And we found some fascinating leads, which will take us to do more work over the next few years. And the lead has been that we found that not only there are abnormalities of the genes, which you read about pretty much almost every week, but what we found is by developing an analytical or mathematical algorithm, we've been able to identify that some of these products of the genes, which are called transcripts, actually connect with each other and form what is called fusion transcripts. And this is a finding that is very novel in the context of breast cancer. And with our analytical tools, we found that some of these fusion genes are seen in repeated tumor samples. So we think the analysis and understanding of the meaning of those fusion genes will allow us to probably develop new diagnostic tools for breast cancer. Number two, potentially identify new targets for drug development, and ultimately help develop new treatments for patients with breast cancer. So that's one part of our work at Mayo Clinic. At the national level, some of the work that we're doing relates to clinical trials, because there's a lot of fascinating information being derived in laboratories, and one of my roles is to try to put this information into the clinic. And we do this through the conduct of these national clinical trials. Cliff alluded to one of these groups, which is called the Alliance, one of essentially 4 National Cancer Institute cooperative groups. So what we do here is that we take the ideas from the laboratory, make decisions related to the appropriate clinical trials to determine whether the information in the laboratory actually can be translated into better treatments and preventive strategies for patients. So I'm very honored to be part of this group, kind of making decisions of what's important at the national level, how can we best conduct these clinical trials to get the answers the quickest potential way for our patients. In the third arena is the international level. Certainly we have to remember that we're part of the entire world. So it is very important as part of my work that some of these ideas for what we call translational clinical trials, and they are these clinical trials in which we also collect blood specimens, tumor specimens, can be done on the global level. Because we should be very honored and proud to be here in the United States, and people around the world really depend on our leadership so that their patients can benefit. So some of my time is devoted to establishing the relationships with investigators in other parts of the world, writing in conjunction with them very innovative clinical trials, and also to monitor those trials on a global basis, be sure that the data that we obtain are reliable data, which then can be translated into what I think will be the new standards of care in the next few years. So as you see, we have the total span from the local work at the clinic, the national work, and then the global work. And I tell you, I'm very proud of our ability to really work as part of a team that has already discovered important things for patients' life. What I'm even more enthusiastic about the future. I think the past was good, the future is going to be great. So thank you very much.